Hey, what's up, everybody? We're going to kick off this episode of Jar Talk with our former head brewer, Matt Hoyle, in just a couple minutes. But I just wanted to log on a bit early to go through how things are going to run and what we're going to talk about. So if you have any questions, post in the comments. Maybe not try and make them not too specific, but more wide-ranging, so it might help other people with the questions that they're going to ask. So I'll just give you a rundown of what we're going to talk about. First, I'm going to ask Matt a bit about his background. I already know that, but so you guys have an idea of where he comes from, how he first learned about kombucha, the different resources that he used, which might be helpful to all of you tuning in who brew kombucha now or who are looking to brew commercial kombucha, or even if you're just brewing at home, some resources for finding out a bit more about commercial brewing or kombucha brewing in general. And then we're going to talk a bit about if you're just starting out. We're going to focus on that for a little while. And that will be the equipment, the instruments you need, health and safety, ingredients, and different resources. Then we're going to talk about sort of a small brewery to a mid-range brewery. What equipment is best to invest in? Do you want a bright tank? Or do you want to continue carbonating in keg? Do you want to force carbonate at all? Do you want to do that naturally? We'll talk about bottling machines, different types of bottling, acidification technology. So we'll dive into the acetator a little bit later. And then basically we're going to talk about brewing methodology, so using a SCOBY versus starter liquid, the benefits and the drawbacks, the ideal measurements for, say, 1,000-liter brew when you're scaling up, ideal temperature, time period, what type of tank, and then a little bit more about carbonation, kegs, bright tank versus natural carbonation. And then alcohol, how to test. Sorry, Matt's just messaging me, so just give me a second here. We're going to talk about alcohol. We're going to talk about how to control it, filtered versus unfiltered kombucha, tips and tricks to control alcohol. Then we're going to talk a little bit about the acetator that Matt designed and built, the benefits of it, the concept, the design, how it works, and then we'll jump into the questions that you guys have. I hope everybody's doing well. If you want to post anything now, you're more than welcome. I can try and answer it in the interim. We're going to try and keep it a little bit lower in time because Matt's got a newborn at home, so we don't want to keep him too late. So if anybody wants to ask any questions now, feel free to post below. And I'll preface this by saying that much of the stuff that I personally know and that we've learned at JAR in terms of scaling up was actually through Matt. Matt came in maybe 2 years into brewing for us, and we were brewing consistently, but it changed the game when Matt got involved. So we're going to log on just now.
Oh.
Hey, dude. How are you?
Yeah, how's it going? Yeah, not too bad.
Good. Thank you for joining me.
Yeah, no worries. Yeah, it's all very random, and this is new to me. I've not done an Instagram live before, unsurprisingly.
Yeah,
So,
it's funny.
it's
I can sort of hear some feedback when I'm talking to you. I'm not sure if that's through your phone.
could be. Yeah, sorry. I was trying to prop it up so that it's not too awkward. It's nearly running out, so it's on charge.
Okay, cool. So, dude, I guess initially I'd like to talk to everybody about your background and how you got to JAR before you came to brewing kombucha.
Yeah, sure. Yeah. I studied architecture in Portsmouth, which is where I'm from originally, and learned a lot and really enjoyed it a lot. The whole kind of creative problem-solving process does suit me, but I realized quite quickly that I didn't want to do the full 7 years, and it was just there's not enough kind of control over the entire product, which is what I kind of enjoy. I like seeing something through from beginning to completion, so which is where having like a working on an actual product is really nice, because you see the entire process and you feel a sense of kind of you feel you've contributed to that thing. Shortly after studying that, I met my girlfriend, who Amy, who we've now had our baby, and she lived in London whilst I was still living in Portsmouth. So, and at the time I'd recently finished studying, so I didn't have a job. So I decided to move up to London and just find what I could. And that started found a job at a chocolate factory and retail shop called Mars Brothers. That's a US company originally. And yeah, started just as a kind of shop assistant there, but then got kind of involved in their production aspects. Like, was kind of curious about how they were doing a lot of things. How they made a range of kind of beverages, and I'd done a little bit of kind of home brewing of wines and beers and stuff like that. Just so had some basis in it. And yeah, through that, eventually sort of progressed up to assistant manager, which was great. Like, it was a fantastic team. The main sort of store manager was excellent. She's an exceptionally good all-round person. A woman called Lani, who's an Australian lady, and yeah, learned an awful lot there. And then it came suddenly to a halt where they decided to close that business, at which point then I had met some of the founders of JAR, not yourself, but Neil, and I think maybe Jess at one point as well, who had come into Mars Brothers and had chatted with them. And I just started reaching out to people because I needed work, and had initially expected to work in the beer brewery because I'd never heard of kombucha at that stage. And then when I came in, I think I met with you on the very first day that I came in, and we sort of talked over that you guys thought that perhaps I'd be better fitted to kombucha rather than beer, and yeah, and just got stuck in from there, I suppose. So yeah.
I often talk about how I can hear myself. I'm gonna see if this. I often there's pre and post. Everything forward.
Yeah.
Brought and understanding previously.
Yeah. Sorry, you're cutting in and out for me. I can't hear all your audio, I'm afraid.
What deal is here? Can you still hear me?
Yeah, I can hear you now. Yeah.
Okay.
You might have feedback, but
Okay. I guess in terms of when you first started, because you didn't know anything about kombucha, how did you learn? Like, what resources did you other than the brewing environment? What did you approach?
That was tricky to begin, certainly at the start, because I mean, initially, obviously, I learned from yourself and from Dom Jones, who was the previous sort of head of production, and yeah, learned the kind of the fundamentals of kind of kombucha, because you guys have been doing it long enough that you understood a fair bit, and you had various books and things around it. But as it kind of progressed, it became kind of clear that there just is a real like dearth of knowledge. There's really not much that kind of tells you certainly how to do it on a large scale. There's an awful lot of information that's good in terms of how to brew kombucha at home and make something that is very make something very simply and that is very tasty. But if you want that thing to be exactly the same every time and to deal with all the challenges that larger scale creates, then it was kind of more looking to like what is kombucha and trying to figure that out, and then also trying to then find like the root knowledge around kombucha rather than necessarily specifically for kombucha, because there are scientific studies. I mean, that's I suppose where a lot of my kind of understanding has really come from, is from places like ResearchGate and Elsevier, I think it's called, and various things that are for sharing of scientific papers. And there's an awful lot of free material out there. Some of it you have to kind of put requests into the authors and things like that. But there is a lot that's dedicated to kombucha specifically, but it's not that kind of coherent. There's an awful lot of knowledge out there that relates to different parts of the kind of kombucha process, but they're not necessarily like synthesized that well. And what you're more looking at is kind of like what is the process that's happening in kombucha, and it's essentially the action of various wild yeasts and various different strains of acetic acid bacteria. That's the kind of fundamentals. I mean, there can be sort of strange exceptions where it's kind of more dominated by lactic acid bacteria and things like that, but then you get into this weird zone where is it kombucha or is it water kefir, and there is a fair bit of overlap, really. It's just depending on which of the dominant organisms. So, but yeah, so it was a range of sources. So yeah, some scientific papers. I had a great book about wine technology that was actually very helpful in terms of a lot of the sort of, because they deal with a lot of the same organisms, because they're considered spoilage organisms in the brewing industry, the wider brewing industry. Yeah, and some, some kombucha based information. So things like I think you had a book by Hannah Crum, which is probably considered a bit of a bible for sort of home brewing, certainly. Mm-hmm. And some of that, there is useful information in there. It's just that, again, at a larger scale, it starts to be lacking. You start to run into things that you didn't necessarily expect or consider. And and also we did early on in my time at JAR, we did bring in a consultant. There was a chap called Will who came in, and I feel like we've since sort of expanded upon some of the things that he referred to, and he had learned about kombucha through a PhD program, I believe, some of his understanding was useful, but I wouldn't say it was complete, certainly in terms of making it a commercial, like industrial process. So it was handy to know things like the cellulose pellicle is not where most of the fermentation happens. It's a component part. You can get rid of it. There are repercussions to that, but yeah, things like that are useful, certainly when you're trying to scale. So yeah, so it's hard to say. A range of sources. I would say look at, if anyone's interested, look at scientific papers based around kind of wild fermentations, very like the kind of yeast that you might expect in bacteria, things like Zygosaccharomyces, Brettanomyces, Schizosaccharomyces, and also just various strains of Acetobacter, Comagata, Aeobacter, Buconobacter, those kind of things. Studies that relate to those, because that's where you find more information about how they actually function and therefore what they are kind of doing during the kombucha process.
Okay, cool. I wish we'd known that before you came along. Yeah. I mean, there's a little bit of echo. I know we're getting some feedback here that says there's echo. So maybe. We were just talking about the brewing.
Does that help a bit if I can just hold?
Let's see. Yeah, there's still feedback. You don't have headphones by chance, do you?
No. Yeah, it's just my phone. Okay. My phone is a bit tired and old, to be honest. So
I mean, we were just talking about sort of the cellulose pellicle versus the starter liquid and brewing that way. Mm-hmm. Can you share a little bit about that process? And
yes, for sure.
How we started doing that at JAR and what the science is behind that?
So when I started at JAR, you guys were using a continuous fermentation process. So that's where you have a static tank that is a pellicle is allowed to form a substantial thick pellicle, and that you draw a certain quantity from it, and then you replace that with sweet tea and just allow the fermentation to progress kind of indefinitely, which again is potentially fine in a like home setup, perhaps. A small volume vessel, you will get quite a reliable fermentation. The fermentation conditions never deviate very much, so it just kind of will eat sugar and produce acid at a sort of set rate. And as long as you're drawing from it reasonably often, then it will always be palatable to drink. Kind of, you just leave it a certain amount of time between each drawdown, and it's fine. The only thing with that is that in an industrial sort of setting, your vessel shape and size is completely different. You're very rarely drawing enough, because we had so many tanks that we needed to draw from and then blend to get a consistent flavor and all this sort of stuff, that often the tanks would sit for too long, and then you'd get all kinds of problems of just it becoming overly acidic, it becoming like off flavors from the activity of yeast, either yeast dying or yeast being kind of stressed and producing all kinds of things like sulfur compounds and stuff that just smell bad and taste bad.
Yeah.
And obviously, the other major factor with kombucha is alcohol. So when you have a pellicle in place, it helps the fermentation in many ways. So it is the natural kind of response of the bacteria to oxygen, and presumably it's an evolutionary response to kind of dominate their environment. So in any kind of fermentation process, what you're trying to do is just create conditions that preference the microbes that you want and the action of the microbes that you want. So rather than just allowing it to be completely wild, and then chances are it won't come out as you expect. Like if you just left some sweet tea with maybe a dash of acid in, chances are it will still go bad. It'll be molds or some other kind of opportunistic pathogen that will get in there, and it will not turn into kombucha spontaneously. So yeah, you're trying to sort of expedite that process and make sure that the end result is like an environment that works for yeast and bacteria and the ones that you would like. So the pellicle does help to do that. So it's created, for anyone who doesn't know, it's created by the bacteria. So many strains of acetic acid bacteria are capable of producing cellulose from glucose predominantly, and they kind of spin it almost how a spider secretes silk. It's kind of like very tiny little fibers. So the cellulose that you're looking at is in the SCOBY, the pellicle, is almost like felt, like microscopic felt, and it's extremely pure cellulose. It's very there's a lot of scientific interest in the cellulose produced by bacteria because it's more pure in form than almost any other form of cellulose in nature. So it has all kinds of interesting properties, but it's very, very absorbent, and it will trap air, trap gases, so the CO2 produced by the yeast, etc. So it helps to make it float, which means that the bacteria are constantly provided a source of oxygen, and also it's like a sponge. So if you imagine in a natural environment where these organisms have come from 100 million years ago or whatever, of like some kind of fruit or some sugary liquid, it's beneficial to create a layer over the top so it stops anything getting in, any other organisms, and it provides, like if the sun's beating down, it's a way of trapping liquid so that they can continue to exist in there, which otherwise it will preventing it evaporating, etc. And presumably it allows them to float to some other place and things like that. It's so in terms of kombucha, that sort of top layer and a little bit beneath it, there's enough dissolved oxygen there for the bacteria to produce acids. Then everything below that is essentially anaerobic. Like there'll be a small amount of oxygen, so it's not fully anaerobic like it would be in a alcoholic fermentation of beer or wine. But it's anaerobic enough that you get more kind of the action of yeast is kind of preferenced. So like the yeast that are found in kombucha generally can also function sort of aerobically or semi-aerobically. They don't mind oxygen, and they will still function like quite well, and they will still produce some alcohol. Many of them also produce acetic acid themselves. Like they're capable of producing acids alongside all kinds of other things. So yeast are far more complicated organisms than the bacteria, and their metabolism can manufacture all sorts of interesting compounds that the bacteria kind of can't really on their own. So lots of kind of esters and higher alcohols and all kinds of things that are flavor compounds essentially. So that's why most alcoholic fermentations, develop complexity. So like wine, what starts as just a fairly simple thing like grape juice becomes this kind of incredible liquid through the action of yeast. So by having that SCOBY there, you allow that to happen. So you get a predominantly kind of anaerobic fermentation and then a little bit of acid formation on top, which means that you'll get a good sort of balance in that your sugar will be consumed fairly quickly. So you get a fairly kind of full fermentation of the sucrose. and you'll get all kinds of nice compounds produced by the yeast. You'll get a tiny bit of natural effervescence, a little bit of, because the yeast is secreting CO2, and then you'll get the acid formed by the bacteria, and the whole kind of combination of some of the acid and the alcohol also kind of is protective in the sense that you are unlikely to see any kind of faults with it. You won't get pathogens coming in, you probably won't get mold growing on the surface, etc. Again, that it comes with the caveat that if you're allowing those yeast and everything to function, they are producing alcohol. So again, that most kombucha, I mean, it depends how much sugar you use, etc. But most that is made on a kind of small scale or a home scale is naturally going to be something like 1.5%, sometimes higher. Like it depends. It depends how it kind of progresses, how much oxygen is available. But yeah, I mean, some go as high as 3% or so, and particularly if you try to sort of secondary ferment in a bottle or whatever, you can see 5% if you're not careful. Mm-hmm. So again, like in an industrial setting, by removing that pellicle, you're allowing more actual dissolved oxygen into the liquid for any kind of given volume, and that means that more of the liquid is able to be utilized by the acetic acid bacteria. So most of the fermentation is actually happening all the way deeper into the liquid anyway. You will still get the same thing where it's predominantly anaerobic towards the bottom, but it helps to speed up the process of the alcohol being consumed by the bacteria and turned into acetic acid mostly. And therefore you will get faster acidification and less alcohol. Again, it's still not a perfect, because again, it's the consumption of alcohol and turning it into acids is again reliant on oxygen. So acetic acid bacteria are obligate aerobes, which means that they have to have oxygen to function. They have to have oxygen to respire, and therefore the rate at which they can consume alcohol is dictated just by how much oxygen you give them. So so as long as there is some alcohol and some other monosaccharides, so things like glucose and fructose present, which then they will produce produce acids rather from that, and so you get just a faster process and quicker acidification. And again, if you're not worried about things like natural effervescence and stuff like that, then and you can't have an high alcohol level, then you have to sacrifice the activity of the yeast to some extent, because you can't have both really. Like the only way you can have both is through strange processes like is it Brew Doctor in the States that uses an actual kind of vacuum distillation system to remove the alcohol, which kind of gives you both, but you will remove other things at the same time. You won't only remove alcohol. It's extremely difficult to only remove alcohol. So
and it costs about $2 million.
Exactly, yeah, it's prohibitive for 99% of kombucha producers. Hopefully that kind of covers it.
That's amazing. Thank you. That's more eloquent detail than anything I could ever share. Well, since we're talking about sort of fermenting without a SCOBY, maybe it's a good time to talk a little bit about the acidification and the acetator, which is, I think, what a lot of people are probably interested in hearing about. Could you explain the process behind that and how you designed it and sort of how it functions?
Yeah, for sure. So the whole kind of idea of it is that it took me a surprisingly long time to cotton onto this, but that kombucha and vinegar are almost indistinguishable, essentially. It's just that vinegar production is done in two separate phases, and with only one end product. So you'll do a strictly alcoholic fermentation with a high alcohol producing yeast species like Saccharomyces cerevisiae, which is used for all brewing, essentially variants of that. You'll produce something that is say 15% ABV, and then you'll hand that liquid over to acetic acid bacteria. And in commercial kind of vinegar production, most of those acetic acid bacteria are actually quite closely related to what you'd find in kombucha. They're not Acetobacter species, they're actually normally Comagata aeobacter species, which kind of are more associated with kombucha generally, but because they can tolerate very high alcohol concentrations and high acid levels. And the way that acetic acid bacteria can then consume that alcohol is through, the rate at which they do it is dictated by how much oxygen is available to them. So alcohol is their food, but in order to eat that food, they need oxygen. So they're like us. Like the only way, we can function sort of anaerobically, but we're predominantly aerobic organisms. We need oxygen to live and for our cells to respire. So in that process, you can go from a 15% ABV liquid, you absolutely saturate it with air to the extent that there's dedicated fermentation tanks called acetators that use a kind of turbine system to almost make the liquid a foam. Practically, there's so many bubbles that you get not only a very high dissolved oxygen, but also an awful lot of kind of surface to liquid interfaces where the oxygen concentration is kind of artificially higher because there's the bacteria can actually sit on the surface of the bubble and consume the alcohol even quicker. And the combination of those two things means that you can get something that's like 15% ABV alcohol turned into 15% acetic acid, because it's pretty much a 1-to-1 conversion in like 36 hours, something like that. So you can make acid extremely quickly if the alcohol is already present, or like if the food is already present for them to do it. So kombucha is different because it's the two things happening simultaneously, and you're not like, it's kind of that the yeast will kind of begin the process by taking the sucrose, secreting invertase, which breaks it down into glucose and fructose, and then they all start to consume that themselves and turn it into various different things, but predominantly ethanol, alcohol. Then the acetic acid bacteria can take that, some of those things, so they can take both the glucose and the fructose that's already that's secreted into liquid, and they can take the alcohol, and most of them will have a bit of a preference for the alcohol, maybe the glucose to some extent, and they'll turn that into organic acids, so the glucose becomes gluconic acid, and the fructose and ethanol becomes acetic acid. And again, there are some other things that they can do. There's some very kind of complicated things. There's a peculiar one which people may have seen if you have very old starter liquid, sometimes younger kombucha as well, which I saw at Jar, is something called dihydroxyacetone that happens like further kind of down the chain, and that is actually the component in self-tanning creams. So it's the same thing. So if you've ever handled kombucha, if you put your hand in some old kombucha and then thought, why is it a bit dark afterwards? It's because you're getting tanned from your kombucha. So there's some weird stuff that goes on.
That was prior to the acetator.
Yes.
Some of our really old stuff that we had in the back part of the room.
Yeah, and there's all kinds of like pigments and weird things. There's I'm trying to even think, melanoids, is it? That other kind of darkening compounds so again, yeah, I mean, we've probably observed it, I don't know if other people have, where if you leave some kombucha to ferment for a very long time in just a static situation, it will eventually become quite a dark color. It goes like it's not colors coming from the tea anymore. It's stuff created by the bacteria. But anyway, I'm digressing. So so in kombucha, you've got those two things happening simultaneously, so it's quite slow. So because generally the sugar available at the beginning is not that high, it's not like it would be in a fruit juice, and it's also sucrose, which is consumed sort of slower, and you've got a kind of generally quite a high stress situation for the organism. So because there's very low nutrients, it's just tea, sugar, and water, and it depends on what sugar you use, etc. But there's not much, and so the whole process happens quite sort of slowly. And I think there isn't a great deal of research out there, but I think there's almost a kind of symbiosis between the two organisms where they both kind of keep each other in check to some extent to make the whole fermentation slower, because it gives them more time to proliferate, and they will kind of continue to have a food source for a longer period of time than if they just ate through it very, very rapidly. Having said that, the sort of again, the limiting factor in terms of certainly the production of the acids, which generally is what you want in kombucha. So most kind of people would define kombucha as being kind of ready when it's acidic, like its sourness is the thing that kind of defines it. And again, that is dictated by how much oxygen is available. It's also dictated by how much food is available. So that's where it becomes slightly more kind of tricky. So, but in the early sort of stages of kind of trying to figure out this stuff, it was just simply seeing what would happen if you provided more oxygen to the bacteria than they get in a static fermentation, which was tried through various ways, but essentially just kind of bubbling air through different methods and trying to sort of agitate the liquid at the same time. And if you simply do that, if you have normal fermenting kombucha and bubble some air through it, you will see an increase in the rate of acidification because you are allowing the bacteria to function just kind of quicker and through more of the liquid than they would naturally. So more of the liquid has enough dissolved oxygen for them to be able to do their thing. What became apparent though is that it was quite erratic. So you would think that just by giving more oxygen, you would get a faster fermentation. And that's not really the case because again, it's reliant on there being a food source for the bacteria. So because the whole fermentation process is quite slow, you're relying on the yeast essentially to produce the food for the bacteria. So you still have to allow that to happen. And if you just simply aerate the whole mixture, the yeast will continue to function, but it's not their kind of preference, and they certainly won't make alcohol very quickly. And alcohol, again, is a kind of ideal food for the bacteria. And so the whole kind of idea with the tank that I eventually kind of made for jar was to actually do more like what vinegar fermentation is, where you kind of split the process into two halves. But you don't separate out the organisms. So because in vinegar you could do it with kombucha, where you have two completely separate fermentations and a sterilization process between the two. But it's just where then you're talking like, is that still kombucha? Would you define that as kombucha? Because technically there was no SCOBY involved, there was no symbiosis. It's just two separate fermentations. So yeah. So but in the tank that we did, essentially it was just creating something where it can initially ferment in an anaerobic way or semi-anaerobic anyway. It's not strictly anaerobic conditions, but enough that there's limited oxygen, so the yeast will function predominantly anaerobically, produce quite a decent volume of alcohol, and then the bacteria can start consuming that quite quickly. So again, because as soon as you give them oxygen through aeration, which is what we did, it was just a kind of a different method of aeration, they then will produce the acid that you want quite quickly because the food is available to them. The caveat with that, and this is something that I learned with vinegar production, through looking at vinegar production rather, is that if you ever see, particularly if you start just aerating kombucha, and you might see a point where there is actually no more acidification, and if in extreme cases, your acid content will actually go down. And what's happening there is that acetic acid bacteria have kind of two ways of existing and sort of respiring. And in the vinegar industry, it's known as acetate overoxidation, which is where the alcohol runs out. So they consume it very quickly. And if the level of it drops below about 0.5%, maybe 0.3, if it gets lower than that, then the bacteria actually start looking for other food sources because it becomes a stressful environment for them. And in many cases, if there's still a high volume of oxygen available, they'll actually start breaking down the acetic acid, so they can utilize that as a food source. So they've produced it, but then they'll start utilizing it, and they'll turn that into essentially water and CO2. So you lose everything you've tried to produce. And I saw that happen at Jar, and so again, it was that you just need to have enough alcohol present at the beginning if you were going to aerate, because otherwise, as soon as that alcohol's gone, the whole process kind of stalls, and you'd have to go back. Once the bacteria have started doing that, they struggle to go back. It's kind of a permanent change to their physiology. So yeah, by simply turning off at the aeration and leaving it for a while is not necessarily going to solve the problem. You have to kind of start again. And yeah, so I think that sort of
Yeah.
explains it. So yeah.
I guess probably for people that are listening or watching, what are ways that people can start doing this on a small scale, without sort of the full picture of like how to put this thing together and all the equipment? What's a good place to start?
Yeah, I mean, I did various different methods at home and at home at Jar rather, and the one of the most effective is just some kind of aquarium pump, like you get a lot of pumps designed for koi carp ponds and things like that, that just to oxygenate water, that are quite energy efficient and cheap, and you don't need high pressure or anything. You don't need an air compressor. Just one of those, and then a fairly hot, like large pore size carbonation stone. So you can get very small ones that are intended for Cornelius kegs and stuff like that. And like a 5 micron maybe sort of pore size on them, because they go down smaller than that, maybe 2 micron. And those work very well to just introduce air into your fermenting liquid, and they will. Yeah, they'll function in much the same way. The only issue with it on that kind of scale is that the cellulose pellicle kind of returns. So the cellulose that the bacteria secrete is in response to oxygen. So when you provide, the reason they do it at the surface of the liquid is because they can tell there's much higher oxygen there, and it's the place they want to be, so they start producing cellulose. But they will do that anywhere if the oxygen is high enough. So if you have a carbonation stone, for example, it varies a lot depending on the conditions in your kombucha and whatever, but generally speaking, within a few days, there will be a fairly defined kind of weird fuzz of cellulose all over it. So it looks completely different to the SCOBY that you'd find on the surface, because it's secreted in kind of long strands, and it's in little pieces that blow around and whatever. But as soon as that kind of happens, it makes the carbonation stone much less effective, because the very fine bubbles that it's producing sort of clump together and just become big bubbles, and it stops working very well. So you have to clean it quite often. It's not terrible. You can normally just pull it off, or if worst case scenario, dunk it in some sodium hydroxide for a while and it will dissolve it. But again, on a larger scale, that's not really practical. So yeah, that's why the tank that I designed used a different system with what's called a Venturi valve, as a way of introducing air but creating conditions where the cellulose formation can't really happen. It could happen sort of within the liquid itself, and so there was a kind of element of strange pieces of cellulose that you do have to kind of filter out in some way, but yeah, it wasn't affected by any kind of clogging, so its performance would remain consistent throughout its time. So yeah, small scale, just aquarium pump, but just be mindful that you'll have to clean the carbonation stone fairly often. If you're using one, you can just use literally just a hose dunked in, but a carbonation stone is a much better use of whatever air you're putting in there.
Or you could spend 40 grand on a Cetotech acetator.
Yes, yeah. I mean, that is the. So I mean, those as I say, they've been designed over many years for vinegar production, and so they're incredibly efficient. Like the mechanism for introducing air is fantastic. But again, in kombucha, you'd almost certainly. Unless you know that your alcohol content is high enough, you'd probably have to turn it off after a few hours, because otherwise you'd have used up all your alcohol that quickly, and then you'd start breaking it down. And you also get some I can't remember exactly why it happens, but if you continue to aerate it at that kind of level, you can get some quite strong bitter compounds produced, and the flavor can be quite unpleasant. So again, you have to be watch for that, because yeah, you don't want to ruin your kombucha.
Yeah, for sure. I guess there was so much I wanted to talk about, but I don't think we're gonna have enough time.
Sorry, I'm rambling.
No, don't worry. This is such important information. I hope everyone's taking notes. But I know a lot of people struggle with alcohol. do you have any simple tips to control your alcohol production? I guess one would be using the liquid starter. Or
Yes.
what are the ways in which you think people can start to control their alcohol a little bit more?
I mean, I think that one of the biggest ones that I seem to have sort of encountered recently is just that an awful lot of people who produce kombucha still do natural carbonation in the bottle or whatever packaging they have, which, you know, again, it can produce something that's quite complex and very tasty, but the consequence of a secondary fermentation that produces carbonation is alcohol. Like, there is no getting away from it, that you're relying on the action of yeast in the bottle in an anaerobic environment. So so that's the biggest thing I would say that a lot of kombucha companies do and will, yeah, you kind of have to move away from, because there's not really any way of doing both. Or you just accept that you're going to have a very tiny amount of carbonation. But yeah, I'd advise against that. Then in terms of your brewing process, yeah, I mean, not having an actual SCOBY, that then it does become a more complicated process, because that SCOBY is your kind of insurance policy to make sure that your kombucha will ferment properly each time, and you're not going to get again unwanted organisms in there. And say if you're starting it with not that much acid at the beginning, so your conditions, say your pH when you start the fermentation is like 5 or something like that, and you remove that pellicle, then you may run into issues. It's hard to say for definite, but you will still, as long as you're using a pellicle, there will be some alcohol that builds up, and as it is being consumed at the same time, as I think I've sort of mentioned before, the general kind of percentage that I'll end up at is about 1.5, something like that. So it depends where you are in the world as to what regulations you have and what you need to worry about, and that may be fine. And so, you know, you don't actually necessarily need to worry. But if you start using like particularly deeper tanks, so if you're having successful fermentations on a small scale, like on your kitchen counter, it's a common assumption, I think, to think that a tank of the same shape will perform in the same way, even if it's much, much larger. But that's not the case, because it's to do with the surface area versus the volume. And the only way, like if you have a jar that's 20 centimeters high, say, and holds, 3 liters, and then you have a tank that's 300 liters, it would also need to be 20 centimeters high to have the same performance. So as soon as you go deeper, you're restricting the amount of oxygen that gets all the way through the liquid. So in that case, it's trying to sort of it just agitate your kombucha in some way, and again, removing the pellicle helps with that, because you're just able, if you regularly stir it to some extent, you're introducing oxygen. Even just simple sort of stirring introduces quite a surprising amount, and again, that will help to just keep down your oxygen, help to break up that strongly anaerobic area towards the base, and then so the way that we kind of settled upon at Jar was that we would start the process with strong starter liquid, which had been produced through aeration ultimately. Prior to that, it wasn't. Like, at one stage we were producing starter liquid just by leaving kombucha for a very long time, which it still works. The only thing is, by doing that liquid will introduce some alcohol. So it's very hard to say, because it depends how old it is as to how much it will introduce. Because eventually, given enough time, starter liquid would have no alcohol in, because it will all get eaten. But that, generally speaking, if you're not fermenting it for that long, you are introducing some from that. And then how much of it you use, if it's fairly young kind of starter liquid, certainly if it still tastes sweet and if it doesn't taste that sour, then chances are it will introduce organisms, and it may be enough to lower the pH, but you're going to have contributed quite a lot of alcohol at the beginning of the fermentation, and then for it to become sour enough for you to package it as you would like, it will again almost certainly create a lot of alcohol. So allowing your starter liquid to be very sour and then not using so much of it, it seemed to be a good way to kind of control that, because by allowing it to become very sour, you do slightly change the microbial balance. So the yeast activity falls off over time, and then it's just a slow work of the bacteria to break down kind of what's left and make acids. So older starter liquid won't contribute as much yeast at the beginning, which can mean that and contributes more bacteria. So you still need the yeast to do what they're going to do, but because there's more bacteria there at the beginning, then the alcohol consumption is quicker. So you can have a more static fermentation that will still not rise quite as high in alcohol, which hopefully that makes sense. So yeah. So again, aerated starter liquid, perfect, because it has another consequence of aeration is that both the yeast and bacteria will increase in number in response to oxygen. So it's a common trick in all kind of brewing industries that at the very beginning of fermentation you will introduce air. So because yeast, in order to make their cell walls, they need some oxygen to produce certain sterols. So yeah, if you don't do that, then you actually have a very sluggish fermentation. So yeah, anaerobic conditions at the beginning at the very start of the ferment aren't good, because the whole process will just be very slow. So but yeah, in an aerated starter liquid, you're allowing that process to happen kind of all the time. So if you compare it under a microscope to old static starter liquid, there's possibly 100-fold more live organisms in there, and so again, it just means that you're kickstarting the fermentation very quickly when you start another one. So yeah.
Thank you, dude. That's incredibly useful. I think for a lot of people. So I'm gonna read out some of these questions that people have now. We'll see if we can get through some of them. Sorry for the echo, but I hope you can hear me. How long do the enzymes and microbes in booch stay viable in a bottle?
Oh, that's a difficult one to answer. So microbes, so enzymes. So there are enzymes. But the primary one really is like the most active one. So again, if your kombucha is only being fed sugar, like any form of sugar, cane sugar, beet sugar, white or whatever, then the main enzyme will be invertase, which is what cleaves sucrose into the two monosaccharides, glucose and fructose, and that will still be in there. That doesn't really go anywhere as far as I'm aware, but its activity will kind of cease fairly quickly. Like generally speaking, like most of the sugar has been broken down within the first week or so. It will continue to sort of rumble on for a while, but not at quite the same rate. So that's hard to answer. In terms of microbes, the bacteria certainly, when there's no oxygen available, they don't actually die very quickly at all. Acetic acid bacteria are quite durable in that sense. They enter a state known as viable non-culturable, I think, where they're still alive, and if you give them oxygen, they'll come back to life, but they're just not doing anything. And I'm not sure I could tell you an exact length of time, because it would probably depend on storage conditions and stuff like that. But a long time, I would guess more than a year for sure, that they would still be alive. Having said that, I don't think that matters. Because
it's actually an interesting question. Is kombucha actually good for you? And if so, why?
So I mean, again, I feel like kombucha there's still research to be done, because it's more complicated than it might first appear. Like the whole kind of fermentation process, because you're using a mixed culture, generally there's very little control over what's in there, and that means that you normally will have a few predominant organisms, but there's all kinds of other ones. And most wild fermentations follow kind of a procession process where different organisms dominate at different stages of the fermentation, because as the conditions change. So what kind of happens at the beginning versus what happens at the end, what might be produced during that sort of time probably varies a lot. But from my understanding, the only possible benefits from kombucha do not come from probiotics. So unless you have a very strange kombucha that is dominated by lactic acid bacteria, which would mean you probably wouldn't see much of a cellulose pellicle, it wouldn't taste vinegary, it would be more akin to a water kefir, then the acetic acid bacteria are not probiotics. They would not be termed probiotic because they do not live within the human body, and they certainly don't confer any health benefits by consuming them. Same with yeast, like, consuming yeast doesn't really, no, as far as I'm aware, has no health benefits besides the like amino acids and proteins and things that yeast themselves contain. What could be beneficial is substances from the tea. So, the tea is known to be kind of good for you, and is pretty much scientifically proven to be good for you. A lot of that comes from various kind of antioxidant compounds like catechins, is that right? And theorubins. Is that the other one that that they break down into? But lots of antioxidant compounds that are kind of known to be good for you. Otherwise, then there's benefits from like the acids themselves. So there's a bit of sort of controversy still because it's hard to kind of prove the levels in kombucha. But there's a particular acid called glucuronic acid that I think is fairly well studied, and can be present in kombucha and can be present in quite large quantities. Like there's some scientific studies where they've managed to make it by modifying the conditions in quite a lot, and that is known to be a kind of detoxifying compound in the sense that it helps to scavenge heavy metals and things from the body, and helps your liver excrete them. So that's potentially good for you. Otherwise, then just acetic acid is certainly within like your kind of mouth and your early digestive tract and things. It does have sort of antibacterial properties for certain organisms. So there's a lot of things that just will be killed quite easily by acetic acid, and so the consumption of it can help to just slightly modulate your microbiome. To my knowledge, kombucha can potentially be good for you. However, in introducing it to like family and friends and stuff, I would tend to say that it's like a soft drink that's not bad for you, perhaps. So because it's just it's something that's nice to drink, but it's quite low sugar content. The sugar that is there is partly broken down, so it's less of a problem than just pure sucrose. And there's some possible health benefits, but my take on kombucha is certainly not that it's any kind of wonder drink. So
okay, that's very very helpful. I also have the same opinion. But a little technical question here from Macarena saying in Mexico, if your starter liquid gets too sour, the pH is too low, how can you pare it back to use it as the liquid SCOBY? Is there any way of bringing it back from being too acidic?
I mean, I don't, in my experience anyway, that's never really been a problem. If it's more acidic and it's stronger, my kind of main tip perhaps with kombucha generally is to start testing your acid content. So rather than just your pH, you want to know how much acid is in your kombucha, because that has much more of an effect on your experience of the drink than pH does. And it's also useful in terms of controlling how much acidity you're carrying forward into a new fermentation through your starter liquid. So from my experience, the more acidic starter liquid is, you may end up with a situation where there's not very many live organisms, but as long as you have dropped the pH enough in your new batch, that doesn't really matter. It might be a bit sluggish at the beginning, but it will take off again. Like, as I say, they're very durable organisms, so they will get going eventually. So I would just say use less of it. Don't unless you find that when you start a new batch, it's like actively not working. Something's not right, and it's not fermenting at all. which I'd be very surprised to see, then just just reduce the quantity that you add, and so to reach the same sort of level of acidity. Hopefully that answers.
Cool. Have you ever tried feeding sugar to kombucha gradually, rather than in one go?
I mean, that is essentially kind of a continuous fermentation process. That's kind of what happens during continuous fermentation, is that you would add a little bit at a time. You take some and then you add some, and you take some and you add some, and so you're introducing fresh sucrose at regular kind of intervals. Having said that, I mean, no, I've not done it from the beginning. I mean, potentially, I suppose. I mean, there's no reason it wouldn't work. It's just you would change the fermentation conditions, because you'd have to be kind of careful in that at the beginning, when there's some sucrose available, that kind of procession of organisms would happen, and then it would start to tail off, and then it would have to happen again and again, right. Potentially. It depends on where in the process you're introducing fresh sugar, but that could, I suppose, have a benefit if you did it very carefully and you were careful. But if you allowed most of the sugar to be consumed, most of the and it's only a small quantity, and then most of the alcohol broken down, you could maybe keep your alcohol levels a little bit lower by doing that. But in my experience, it seems that some sucrose is always kind of left. It takes a very, very long time for it to be completely broken down, because the fermentation is quite slow, and therefore I'm not sure that you would see any particular benefit from that. It's worth experimenting with, though.
Yeah. Okay, cool. There's one question here about what is the ideal total acidity, pH, and Brix of a finished starter liquid?
That's a hard one to say. Because I'm not sure that there is such thing as a finished starter liquid, because again, if you kind of refer back to vinegar, you could say that is a fully fermented product, in the sense that you've taken your initial sugars, you've turned those completely to alcohol. There's no sugars remaining. Then you take that and turn that completely to acid. So there's only a tiny amount of alcohol remaining, and that's essentially it. Kombucha is not really like that, in that for that to happen, for it to become fully fermented, so that everything has broken down and become only end products, technically would actually be at the point where the bacteria are breaking down the acetic acid, and you'd end up with something that's basically just kind of water again. So I'm not sure I'd say that there's a finished one, but generally speaking, you just want a high level of acidity, so that you can not use very much of it, and so that it will introduce, it will lower your pH well, and it will introduce the kind of organisms you want, predominantly more bacteria versus yeast. And in my experience, that kind of happens. Like, a good level is something like 0.3 to 0.4 moles per liter, which as a kind of percentage is probably about 1.5 to 2% acid. I use the expression moles per liter because kombucha contains a mixture of acids, so you can't say exactly what those acids are, like what the balance of them is, and therefore you can't say it is a percentage of this acid, because they have, if you measure it through titratable acidity, which is what I'm kind of talking about, you can't then infer from that what percentage of acid is present. So but as a rough guide, if you use acetic acid as your base, then it's something like 1.5, maybe 2%, and that will work well. In terms of Brix and stuff, again, that doesn't particularly matter. Again, in my experience, it's good to start quite high. If your goal is to make starter liquid, you want to just provide plenty of sugar so that the process can happen. So I would normally start a starter liquid at something like 10 Brix, so 100 grams per liter of sugar. And then allow that to carry on. And the pH during that time can go as low as like 2.2, something like that. It can go very, very low. But as long as it's sort of under 3, I'd say it's okay.
Mm-hmm. Okay, cool.
Okay, cool.
Awesome. We just have one more question. I mean, there's quite a few, but there's two of the same question, which is how do you determine the expiry date? Is that generally a flavor thing, do you find? Or an alcohol production thing? Oh shit, we only have 10 seconds remaining.
Expiry date. It doesn't. Yeah, flavor. I think is all that you can go by.
Wait,
it doesn't. Kombucha doesn't really go bad. So
I was just gonna ask. Cut us off. Hold on.